plant hormones like gibberellin, cytokinin, and ethylene. Okay, yesterday we discussed the auxin. Okay, after molecular basis of inheritance, we discussed the three hormones. Okay, now we discuss the what molecular basis of inheritance. In that, in this chapter, nucleosome is important, genetic code is important, lac operon is important, Grift experiment is important, and DNA replication semi-conservative experimentally proved by the Michelson and Stahl. These are the important. Okay. Now we discuss the what? First, lac operon. First one is lac operon. Lac operon. Okay. These are which chapter? Molecular basis of inheritance. Molecular basis of inheritance. Okay, in this chapter we discuss the lac operon, and the next one is what? A nucleosome. Lac operon, nucleosome, and then after nucleosome, the next one is what? Transformation principle. Transformation principle. Okay, then next one is what? DNA replication semi-conservative. Experimentally proved by the what? Michelson and Stahl. Michelson and Stahl experiment. Okay. And the next one is what? Genetic code. It's a very important genetic code. Okay. The next one is what? Difference between DNA and RNA is important. Difference between DNA and RNA is important. Now we discuss the one by one. First, lac operon. Okay. In these 5 questions, compulsory appear the what? One question in your board level. Lac operon. Lac is nothing but lactose. Lac is nothing but lactose. What is the operon? Operon is nothing but structural gene and functional genes. Both are fit together, both are tied together on the where? On the DNA segment. On the DNA segment, it's called what? Operon. Okay, lac is nothing but lactose. Operon means both structural and functional genes are tied together or fit together on the DNA segment. It's called what? Operon. Okay. Now, these are the bacteria. These are the what? Bacteria. These bacteria, in this bacteria, we give the water lactose. We apply the water lactose. We give the water lactose to the bacteria. Okay, what happened to their metabolism? Or in the absence of these are what? Bacteria. Okay, in the absence of bacteria, we are not gives the word lactose. What happened in their metabolism? These are what? These black colored dots are lactose. These black colored dots are lactose. In the presence of lactose, what happened in their metabolism? In the absence of lactose, okay, what happened in their metabolism? Now we discussed. Okay, now these are first one. What are structural genes? Structural genes are Z, Y, A. Structural gene. Okay. Now functional genes are regulatory genes. Functional genes are regulatory genes. Functional genes are regulatory genes are PEPO. P promoter, inhibitor, again promoter and operator. PEPO. Okay. These are what? Regulatory genes are functional genes. These three are what? These three are ZYA is what? Functional genes. Sorry, ZYA is what? Structural genes. Okay. Now, first one. These are DNA segment. These are DNA segment. These are DNA segment. Okay. In the DNA segment, first one, what it's called? Functional genes are there. Functional genes like PEPO, PIPO. Okay. Then next is what? Structural genes. Z, Y, A. These three are what? Structural genes. These three are what? structural genes. Structural genes. These three are what? Sorry, these are what? These three. Which one? P for promoter, I for inhibitor, again O for operator. P promoter, I inhibitor, again P promoter, O operator. Okay. Then now, in the absence of lactose, in the absence of lactose, what happened? In the absence of lactose, what happened in the bacteria? Okay. Like DNA depends. These are DNA segment now. These are DNA segment. These are what? Which segment? These are DNA segment. Okay. Now from the DNA, RNA is formed. It's called transcription. You know already that. Say DNA depends. Which enzyme? DNA depends. RNA polymerase enzyme binds to the water promoter region. Which enzyme? DNA depends. DNA depends. RNA polymerase enzyme. DNA depends. RNA polymerase enzyme. Bind at a promoter region. Bind at promoter region. Now what happened? This promoter shift to the water, that enzyme from this region to this region. Okay, they are shift from promoter to inhibitor region. Now in the inhibitor, in the presence of what? DNA depends RNA polymerase enzyme undergoes transcription. Undergoes what? Transcription, and form a what? mRNA is formed. It's formed the what? mRNA is formed. Okay. Which mRNA? Repressor mRNA. Repressor mRNA. Okay. During the translation, now during the translation, during the translation, this repressor mRNA forms a what? Protein. Okay, formation of mRNA into protein, it's called what? Translation. Now okay, now from repressor mRNA to which protein is formed? Repressor protein is formed. Repressor protein is formed. Now this repressor protein is active. Okay, repressor protein is active. Now this active form of repressor protein bind to the active form of repressor protein bind to the what? Operator region. Active form of repressor protein bind to the what? Operator region, and their function is switch off at the operator region. Now ZYA gene does not going on their function. Okay, so these are what? Absence of lactose. What happened in the absence of lactose? Okay, in the absence of lactose, first one, DNA depends RNA polymerase enzyme. RNA polymerase enzyme. DNA depends RNA polymerase enzyme bind at the promoter region. Bind at the promoter region. Bind at the promoter region. Now from promoter to it shifts to operator region. Sorry, not operator, inhibitor region. Inhibitor, shift to inhibitor region. Now from at the inhibitor, what happened? At the inhibitor, okay, from DNA to RNA is formed. Okay, which RNA? Repressor RNA. Which RNA? Repressor RNA. Okay, this process is called what? Transcription. This process is called what? Transcription. Okay, then now from repressor mRNA to from repressor mRNA to repressor protein is formed. Repressor protein is formed. Repressor protein is formed. Okay. Repressor protein is formed. Is called, this process is called, this process is called what? Translation. Is called what? A translation. This process is called what? A translation. Okay. Now repressor protein bind to the repressor protein bind to what? Repressor protein bind to the operator region. Okay. Repressor protein binds to the operator region and their function is stop. Their function is what? Stop. Okay. That's why what happened that Z gene, Y gene and A gene do not involve in the function. Okay. These are about absence of lactose. Now presence of lactose, what happened? Now presence of lactose, what happened? Okay. Same. Okay, same points. Like these are DNA segment. DNA segment. On the DNA segment, what? Structural gene and functional genes. Okay. Now these are functional genes. PIPO, PIPO. These are what? Functional genes or regulatory genes. And ZYA. ZYA is what? Structural genes. Okay. These are presence of lactose. These are presence of lactose. Okay. Same pointer. DNA depends RNA polymerase enzyme binds to the promoter region. Now promoter to it shift to the what? Inhibitor region. Okay. Now this inhibitor, what happened to undergoes? This I gene is undergoes what? A transcription. I gene undergoes transcription and form a what? A repressor mRNA. It's formed the what? A repressor mRNA. Okay. From repressor mRNA to undergoes translation. Okay. Repressor mRNA undergoes translation, form a what? A repressor protein. Okay. Now these are presence of lactose. Now these are what? A lactose. These are what? A lactose. Lactose also called inducer. Lactose also called what? A inducer. Okay. Now this lactose bind to the repressor protein. Now lactose bind to the what? A repressor protein. These are lactose. This lactose bind to the what? A repressor protein. Now these are what? A repressor protein. Now these are repressor protein bind to the what? A lactose. So in the presence of lactose or after binding the what? A lactose to what? A repressor protein. What happened? It's inactive. It's a inactive. So inactive form of repressor protein does not bind to the operator region. Okay. What? A inactive form of repressor protein does not bind to the what? A operator region. Now if does not bind, this Z gene, Y gene and A gene is code to the what? A different functions. Means it's code to the what? A different like Z gene code to the what? Z gene code to the what? Beta galactosidase enzyme. Y gene is code to the permease enzyme. A gene is code to the trans-acetylase enzyme. Now we discuss that. Z gene code to the what? Beta galactosidase enzyme. Z gene code to the beta galactosidase enzyme. Okay. Now Y gene is code to the permease enzyme. And then A gene is code to the trans-acetylase enzyme. Okay. Now what is the function of this beta galactosidase enzyme? This beta galactosidase enzyme break down the water lactose. Break down the water lactose. Lactose is a disaccharide. Disaccharide means nothing but di means two. Saccharides. Saccharides means nothing but sugar units. Sugar units means these two sugar units. Okay, lactose has how many sugar units? Two sugar units. Now this lactose breakdown in the presence of beta galactosidase enzyme and form a what? Two monosaccharides. Form a what? Two monosaccharides. Okay. So Z gene go to the beta galactosidase enzyme. This beta galactosidase enzyme break down or hydrolysis. Breakdown or hydrolysis. Okay. Hydrolysis of lactose to galactose plus glucose. Okay. Then next one, Y gene is code to the what? Permease enzyme. Okay. After breakdown of lactose into galactose and glucose, now this permease enzyme easily diffuse the water glucose into the cell. Easily, permease enzyme helps the water permeable. Permease is nothing but permeable. Permeable is nothing but allow. Okay. So this is a permease enzyme allow the water glucose into the what? Bacterial cell. And then A gene put the water transacetylase. Okay. After entering the what? Glucose into the bacteria, and that transacetylation reaction occurs in the where? In the bacteria. These are what? Presence of lactose and absence of lactose. Okay. What happened in the presence of lactose? In the presence of lactose, this repressor protein bind with the water lactose. This lactose also called allosteric lactose also. Okay. And these are also called inducer. Okay. This inducer bind to the repressor protein. In the presence of inducer, this repressor protein is inactive. So inactive form of repressor protein does not bind to the operator region and their function is going on. Okay. So Z gene code to the beta galactoside, Y gene is code to the permease, A gene is code to the transacetylase. Okay, Z gene code to the beta galactosidase enzyme. This beta galactoside enzyme hydrolyzes the lactose into galactose and glucose, and Y gene is permeable the glucose into the cell, bacterial cell, and a transacetylase is coded by the what? A gene. Okay, so this transacetylation reaction occurs in the where? In the bacterial cell. Okay, these are about presence of lactose. The next question is nucleosome. Second question is nucleosome. When observe this chromatin material under the electron microscope, this nucleosome appear a what? Beads on strings. Beads on strings. Okay. When nucleosome, not nucleosome, chromatin. When chromatin observe under microscope. Which microscope? Electron microscope. Electron microscope. What happened? It's look like a beads on strings. Beads on strings. Here beads are nothing but nucleosome. Beads are nothing but nucleosome. Strings are nothing but chromatin. Strings are nothing but chromatin. Okay. Right. Now these are bead. Bead is nothing but nucleosome. These are bead-like structure. Okay. Then on the bead have what? Histone proteins. On the bead have what? 8 histone proteins are there. 8 histone proteins. Okay. There are H2A, H2B, H3 and H4. All are pairs. So 4 types of histone proteins are pairs. So 4 into 2 equal to 8. It's called octamer. It's called what a octamer. 8 histone proteins are there now. That's why what it's called octamer. Now H2A and H2A. Okay, then H2B and then H2B, then H3, then H3, then H4 and then H4. 8 proteins. There are what 4 types. Okay, outside of the nucleosome. These are nucleosome now. In the nucleosome has a 8 histone proteins. Outside of the nucleosome has H1 protein. Outside of the nucleosome has what a H1 protein. Histone 1 protein. Okay. Now histone proteins are These histone proteins are positive in charge. Histone proteins are positive in charged because of they are derived from basic amino acid. This is because these histone proteins are derived from because of it's derived. They're derived from basic amino acid. They're derived from basic amino acid. Lysine and arginine. Lysine and arginine derived from lysine and arginine. These are what basic. That's why these are positive in charge. Histone proteins are what positive in charge. Okay. Now on this histone proteins are wrapped by the DNA. Nothing but chromatin. Histone proteins are wrapped by the what DNA. So see, these are what chromatin. This chromatin wrapped the what histone in 2 turns. From here to it start the what it begins from here and end with the end here. So it's a 1 turn, and again it start from here and end with here. So it's a how many turns? Are 2 turns. So histone proteins wrapped by the DNA in how many turns? Are 2 turns. DNA or chromatin wrapped to the what? To the histone proteins. DNA wrapped to the histone proteins in how many turns? In 2 turns. In 2 turns. Okay. 2 turns. Okay. Then now this DNA is a negative in charge and the histone proteins are positive in charge. So positive and negative both are what attractor. Then now this chromatin is tightly bind to the what histone protein. Sorry, H1 protein. These are tightly bind to the what histone H1 protein. Again, other nucleosomes are repeated. This nucleosome also consists of what 8 histone proteins, and these chromatins wrapped the what the histone protein in 2 turns continuously. Again, H1 is protein. H1 protein is bind to the what the histones are continuously. Okay, and then wrap this DNA wrapped to the what nucleosome. Now that nucleosome wrapped DNA consists nucleosome DNA consists 200 base pairs. 200 base pairs. Okay. Now chromatin is nothing but this DNA and then nucleosome is called what chromatin. These are about a nucleosome, and lactoferrin also completed. Then next one, transformation. Third one is transformation. Third question. Transformation. Okay. Frederick Griff experimented on Streptococcus pneumonia bacteria. Okay, Frederick Griff experimented on which bacteria? Streptococcus pneumonia bacteria. Streptococcus pneumonia, P is silent here. Streptococcus pneumonia bacteria. Streptococcus pneumonia bacteria. It caused the which disease? This bacteria caused the which disease? Pneumonia disease. It caused the pneumonia disease. It caused the pneumonia disease. Okay. Then now he cultured there were two strains of bacteria. He was cultured there were two strains of bacteria. Two strains of bacteria. Okay. Strain 1 is a smooth surface. And a strain 2 is a rough surface. Strain 2 is a rough surface. Okay. In short form, it's called S strain. Starting letter is S now. That's why in short form it's called what? S strain. In here, in short form, it's called R strain. R strain. Okay. Now this smooth surface means S strain cause the disease. S strain cause the disease. That's why it's a virulent. Okay. R strain does not cause the disease. That's why it's a virulent. A means absent. Which one is absent? Disease causing agent is absent. That's why it's called what? It's called a virulent. A virulent. Okay. Now in four step we can explain. Step 1. S strain bacteria. Yes, sir. Strain bacteria inject into the mice. What happened? That mice will be die. Because of S surface means nothing but a smooth surface is a virulent. This virulent bacteria inject into the mice. It causes the disease now. That's why that mice will be die. Okay. Then in second step, R strain inject into the mice. That bacteria is live. Because of this R strain is a virulent, does not cause the disease. That's why that bacteria is a live. Then third step, mixture of heat killed S strain bacteria. Inject into the mice. Mice is alive, not die. Mice is alive because of that disease causing agent killed by the heat. Heat. So that this disease causing agent property is destroyed. That's why not cause the disease. So mice will be live. Then next one is what? A mixture of disease causing. A mixture of S strain and a live R strain. A mixture of heat killed S strain. A mixture of heat killed S strain. A mixture of heat killed S strain and a live R strain. Inject into the mice. Mice will be die. Because of when this bacteria is heated. Okay, what happened? Some substance are transmitted from S strain to R strain. Okay, some substance are transferred from S strain to, these are what? S strain substance. Okay, these S strain substance are transported from S to R. Now this S strain transmitted to R. Now this S strain substance synthesize the what? Disease causing agent in the R strain. That's why R becomes the what here? S. S. That's why smooth surface cause the what? Disease. That's why that bacteria will be die. So here transfer the what? Substance from S strain to R strain. Transformation is occurs. That's why this principle is called what? Transformation principle. Discovered by the Griffith in Streptococcus pneumonia. The next one is difference between DNA and RNA is also important. Difference between DNA and RNA. difference between difference between DNA and RNA. DNA and RNA. Okay. Now here the first column DNA and second column RNA. So what is the abbreviation of DNA? Deoxyribose nucleic acid. Deoxyribose nucleic acid. RNA, ribose nucleic acid. Ribose nucleic acid. Okay. In DNA has how many strands? DNA has double stranded. Say double helical or double helix. Double helical or double helix. Double helical structure. Double helical structure or double stranded or double strand. In here has only one strand. Only one strand. Okay. The next one in the DNA, which one is the sugar in the DNA? Deoxyribose sugar. Deoxyribose sugar. Deoxyribose sugar in the DNA. In the DNA, deoxyribose sugar is a sugar. Is a sugar in the DNA. Here ribose sugar in the RNA. Ribose sugar in the RNA. Okay. Now DNA act as a genetic material. DNA act as a genetic material. Genetic in material in most of the organism. In a most of the organism. Most of the organism. Now RNA act as a genetic material in some viruses. RNA not only act as a genetic material, not only act as a genetic material. Okay. Genetic material in where? In viruses. In a virus also perform, not only genetic material, also perform the what other function. Also perform the what? Also perform the what? Other function. Other function. Nothing but first one structural and a catalytic, and then adapter, and then adapter. Okay, etc. The next one, DNA does not directly synthesize the protein. DNA depends on the RNA for synthesis of protein. Okay, DNA like this. Central dogma, you know central dogma. These are also important for 2 marks. Central dogma discovered by Watson and Crick. Okay, so what is the central dogma? Formation of DNA to mRNA. Formation of DNA to mRNA. This process called transcription. Transcription. Okay. Now mRNA to protein formation is called what? Translation. Translation. Okay. These are called central dogma. See, this DNA depends on what? This DNA does not directly synthesize the protein. This DNA depends on the RNA. Then RNA synthesize the protein. So for the protein synthesis, DNA depends on RNA. This DNA does not synthesize directly the water protein. Okay, DNA does not directly synthesize. DNA does not directly synthesize the proteins. Okay, these are depends on RNA for protein synthesis. For protein synthesis. Okay. Then RNA is directly synthesize the proteins. RNA is directly synthesize the proteins. Okay. The next one, DNA is stable. structurally and chemically stable. Okay. DNA is structurally and chemically stable because of thymine is there, and at the third carbon have a hydroxyl group. That's why DNA is a structurally and chemically stable because of at the third carbon OH group, and then thymine is present, okay. But RNA is not stable. RNA is a not stable because of additional OH group at the second carbon, and then what uracil is present in that. RNA does not stable. RNA does not stable because of what? Because of additional OH group is present. Additional OH group is present. Additional OH group is present at a second carbon. At second carbon, and then next one is what? Uracil is present in that. Uracil is present. The next point, DNA is mutant slowly. In DNA, mutation occurs slowly because of stable. DNA mutation occurs. In DNA, mutation occurs slowly. In DNA, mutation occurs slowly. What is the mutation? Changes. Okay. In RNA, mutation occurs rapidly. In RNA, mutation occurs rapidly. Okay. Then now, which nitrogen base are present in that? Which nitrogen base are present in the DNA? Okay. Nitrogen bases are adenine, thymine, guanine, and cytosine. Okay. Adenine, guanine, thymine, and cytosine. In between the AT double bond, comma, in between the GC triple bond. Okay. Then now, in here, thymine is absent, right? So in RNA, nitrogen bases are RNA nitrogen bases are adenine, uracil, guanine, and cytosine. In between the AU double bond, in between the GC triple bond. Okay, DNA is only one type. Of genetic material. DNA is a only one type, but RNA is a three types. RNA is a three types, but what's that? Okay, tRNA, okay, mRNA, and then rRNA. These are what? Three types. These are about the difference between DNA and RNA. The next one, genetic code, and then tRNA structure also important. Okay. Genetic code. Okay. In genetic code, we have what are 20 amino acid. We have what are 20 amino acid. The 20 amino acids are synthesize the proteins. 20 amino acids synthesize the proteins. Okay. Then in the RNA, in the RNA has a how many nitrogen bases? 4 nitrogen bases. 4 nitrogen bases. What that? Adenine, uracil, guanine, cytosine. Okay. Now the 20 amino acids code to the water these 4 nitrogen bases. How? See, for example, this how many nitrogen bases? 4 now. 4 nitrogen bases are code to the what? The 20 amino acid. 4 nitrogen bases. 4 nitrogen bases code to the what? The 20 amino acids. For example, if these 4 nitrogen bases code to the what? The 20 amino acids. If one codon, if one letter, so how many codons are formed? Only 4 codons are formed. 4 codons are formed if code to the one letter. If code to the one letter, one letter, how many codons are formed? 4 codons. So remaining 16 amino acids are does not code. Remaining 16 amino acids are does not code. Then next one, if that nitrogen bases code to this amino acid to the 2 codons, 2 letters, not 2 codons, 2 letters. So how many codons are formed? 4 into 4. 4 into 4 means 16 codons are formed, right? Remaining how many amino acids are does not code? 4 amino acid does not code. 4 amino acid does not code. For example, this amino acid code to the what? 3 letters. 3 letters means 4 into 4 into 4. 4 4 is 8. 8 4 is 64. Okay. Sorry, not 8 4 is 64. 4 into 4. 4 4 is 16. 16 4 is 64. Okay. So 64 codons are formed. 64 codons are formed if code to the 3 letters. If code to the 3 letters, it's formed what? 64 codons. Okay. 64 codons. So here codons are more and amino acids are what? Only 20 amino acids. Means number of codons are code to the amino acids. Okay. Out of 64 codons, 61 codons are code. 61 codons are code to the amino acid. Code to the amino acid with 3 letters. With 3 letters. So that's why what it's called triplet codon. Triplet codons. Okay. Remaining 3 codons. So total 64 codons. Out of 64, 61 codons are code to the amino acid. Remaining 3 codons are does not code to the amino acid. That 3 codons are called. 3 codons are called stop codons. 3 codons are called what? Stop codons or nonsense codons. Or nonsense codons. The name is UAA, UGA, UGA, UAG. UAA, UGA, UAG. Okay, these 3 are what? Stop codons. Then now, what are general features of genetic code? Salient features of genetic code. Okay. First one, codons are degenerate. First one is codons are degenerate or unambiguous. Codons are unambiguous. Point 1. Salient features of genetic code. Codons are unambiguous. Codons are unambiguous. Codons are unambiguous means one codon code to the only one amino acid. That codon does not code to the what another amino acid. For example, U is there. This codon code to the only phenyl amino acid. Phenyl alanine amino acid. Phenylalanine amino acid does not code to the any other amino acid. Does not code to the any other amino acid. That's why what it's called unambiguous. Okay. Then next one is what? The next one, codons are degenerate. Second feature is what? Codons are degenerate. Means one amino acid code more than one codon. One amino acid. Okay, one amino acid code more than one codon. One amino acid code more than one codon. Like glycine, alanine, valine. Example, glycine, alanine, okay, valine. These are the amino acid. These amino acid code more than one codon. Serine also. Okay. Then next one, codons are universal. Next one is what? Codons are universal. Codons are universal. For example, in prokaryotic cell 2, multicellular eukaryotes. Prokaryotic cell 2, eukaryotic cell. Okay. For example, in prokaryotic cell, triple U also code to the phenylalanine, and in eukaryotic cell, triple U also code to the what? Phenylalanine. So that's why these are what? Universal. Exception in a mitochondria and protozoans. Okay. Exception in mitochondria. Exception in mitochondria and protozoans. Okay, mitochondria. Then next one is what? These are the third general feature, salient feature. Fourth one is codons are continuous. When we read the mRNA, like these are the mRNA, and the mRNA have what? Codons like this. AUG is there, GCA is there. Okay, then AUC is there, and GAG is there. Okay, when we read the word this amino acid on the mRNA, does not give the what? Commas and then full stop. Okay, so it's read continuously. Does not give the what? Does not give the commas. Okay, and full stops. Say continuous. It's a continuous. Okay, then fifth point, methionine involving dual role. Methionine is a perform dual role. Methionine perform the what? Dual role. One is code to the what? AUG. Methionine is a initiation of translation. Okay, AUG is code to the what? Methionine. AUG is code to the what? Methionine. This methionine initiate the translation. Also code to the what? Methionine. These are what? Salient features of genetic code. The next one. So Griffith experiment is over. Lac-Operon is over. DNA, difference between DNA and RNA is over. Genetic code and nucleosome. These are completed. The next one. Gibberellins. You can go to the what? Plant growth and development. Sorry, before that, tRNA structure is important. tRNA structure. tRNA. tRNA is a clover leaf-like structure. Clover leaf-like structure. Are soluble RNA, or also called soluble RNA. Okay. And then next one, these are inverted L shape. These are inverted L shape. Inverted L shape. Okay. Say these are 5' end. These are 3' end. Okay. Now these are arms. These are what? Arms. These are also arm. These are loop. These are what? Loop. Okay. These are also arm and these are the loop. Okay. These are vestigial arm. Then these are what again? Arm. Okay. These are what? A loop. Okay. Then now these are what? Anticodon stem. Okay. Anticodon. These are anticodon loop. These are anticodon loop. These are anticodon stem. Anticodon stem. Okay. These are amino acid binding site. Amino acid binding site. Okay. Then these are what? Ribosome recognized site. These are only ribosome recognized site. Okay. In our exam, given the what way, find out the 5' end, find out the what 3' end or amino acid binding site or anticodon stem. Okay, and anticodon loop. Okay. These are what? See, these are what? Inverted L shape, like this. See, inverted L. Okay, like this. Inverted L shape. Okay, inverted L shape. And these are soluble in the where? In the sodium chloride. See, 0.5 sodium chloride in 0.5. So, dissolved in the where? In the sodium chloride. That's why it's called what? A soluble RNA. And then these are what? Like a clover leaf like structure. Okay, these are about tRNA. The next, gibberellin. Gibberellin. These are plant growth and development chapter. Plant growth and development. Plant growth and development chapter. Okay. Now gibberellin. So hundreds of gibberellins are reported in where? In higher organisms and fungus. Okay. What are hundreds of gibberellins are reported. Hundreds of gibberellins are reported in where? In a higher organism. In a higher organisms and fungus. And then what are the names? GA1 means gibberellic acid 1, GA2 gibberellic acid 2, comma GA3 gibberellic acid 3, and then GA100 gibberellic acid 100. Hundreds of gibberellic acids are discovered. Among that, GA3 was a first discovered. GA3 is a first discovered. Okay. And gibberellic acid is which in nature? These are acidic in nature. Okay. Now physiological roles of gibberellic acid. Physiological role of gibberellic acid. Physiological role of gibberellic acid. First, gibberellic acid increase the water length of axis. Means when gibberellic acid spray on where? On the grape stalk. On the grape stalk. What happened like this? These are the grape. These are what the grape stalk. When we spray the gibberellic acid on the grape stalk, what happened? They increase the length. Gibberellic acid spray on the grape stalk. What happened? Increase their length. Increase their length. Increase their length. Okay. The next point is what? Next point. It's improve the what? Elongation and then improve the shape of apple. When gibberellic acid spray on the apple, it's improve their shape. Improve the shape. Improve the shape and elongation. And elongation in where? In a apple. In a apple. Then next one is what? Gibberellic acid speed up the what? Malting process. Gibberellic acid GA speed up. Gibberellic acid speed up the malting process. Gibberellic acid speed up the malting process. Okay. The next fourth role. Generally, sugarcane store the carbon in the form of sugars. Sugarcane store the carbohydrates. Sugarcane store the carbohydrates in the form of sugars. In the form of sugar. When we spray the gibberellic acid on the sugar, what happened? The sugar yielding is increased 20 tons in per acre. Okay. Sugarcane crop. Okay. Sugarcane crop. Sugarcane SA means nothing but sugarcane crop is increased 20 tons per acre. 20 tons per acre. 20 tons per acre. Then next one is what? It's when we spray the gibberellic acid in the juvenile conifers, what happened? Early seed production is occurs. Okay. Spray the gibberellic acid on juvenile conifers. Okay, spray the gibberellic acid. Spray the gibberellic acid on juvenile conifers. On a juvenile conifers. What happened? What happened is early seed production. Early seed production is occurs. The next one, gibberellic acid also helps in where? In bolting process. These are the important all in 2 marks also. Bolting mostly seen in the where? In the rosette manner of leaves. Okay, means in beet and cabbage. In beet and cabbage. When gibberellic acid spray on, sorry, not what is the bolting first? It promotes the what? Bolting process. Gibberellic acid promotes the what? Bolting process. What is the bolting process? Elongation of internode. When just prior the what? Flowering or reproductive stage is called what? Bolting. When we spray the gibberellic acid to the beet or cabbage, what happened? Increase their what? Or elongation of their internode. In which condition these are elongated? Prior means before the flowering. Okay. Bolting means increase the what? Or elongation of what? Internode. Elongation of internode. Just prior to reproductive stage. Just prior to reproductive stage is called what? It's called what? Bolting. These are about what? Gibberellin. And also delay the senescence. Gibberellic acid also delay the senescence. That's why that fruit can extended on the tree still in the market period. Okay, these are about gibberellic acid. Then tomorrow we discuss the ethylene, and only ethylene is there, and cytokinin. After cytokinin ethylene, then goes to the what? Genetics. If genetic is completed, then all SAQs are completed. Okay, then we start the VSAQs. Okay, mostly 40 marks questions are 40 marks answer is completed. LAQs are completed, and very important SAQs are also completed. Then remaining only VSAQs are there. Okay.